Welcome, 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 grade 10s to yet another awesome life science edition. I hope that you are all very, very well. And today you're going to see we're going to start with exam preparation. Most of you are going to be starting exams in the next couple of weeks. So I hope that you have a study timetable, that you have allocated times in which you need to study. I know some of you are laughing and going, ha, 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 right? But if you really want to do well, you need to start prepping for exams, for life science especially. I hope that you have started to do your summaries. Right, so it's very difficult to study from a textbook. All tests have proved that the best way to study is by your own handwriting. And that's not by writing out. I always suggest to mine, you get a nice big A3 piece of paper, right? Lots of pictures, lots of colorful notes, and then you use your textbook just to get those little nitty-gritty facts out. Okay, so today, not too much of a lesson. We are going to practice, all right, certain questions that might come up in your exam paper. If you have a look on the screen in front of you, right, you will see what we're going to cover this week, and then we'll cover the rest of the topics next week. So if we're looking at, it's all work that we've done from the beginning of the year. So you're going to have to, all right, take all the work. You need to study everything that you have studied from the beginning of the year, and you now need to, obviously, in a nice big exam, you're going to need to reproduce all of that. Some of you might write one paper, some of you might write two papers, and this week, you see, we're going to cover biochemistry, right? We're going to look at there, we're going to cover cells, mitosis, mammalian tissue, and plant tissue. And then next week, we're going to do, all right, all the plant organs and the transport and the skeleton. So make sure that you have all of your resources handy, and for each of those, I do suggest that you do have summaries that you can go through, and that you're not trying to study volumes and volumes and volumes of work, right, that is in your textbook. Now the first question I'm going to give you to do, because it's a nice opening question, right, and it's a question on the cells. So I'm going to give you 3 minutes, right? 3 minutes, just a bit of a starter to get everything up and going. I'm going to flash the questions up on the board. There's a diagram, right? If you see, there are two cells with their organelles, and the questions are as follows. Let me go through them with you quickly, and then we'll come. Which cell, X or Y, represents a plant cell? Number 2, give 2 visible reasons for your answer. Number 3, label parts A and B. Number 4, structure C plays an important role in cell Y. There's 3 functions of the structure. And number 5, name the organic substance that part D in cell Y mainly consists of. Okay, just a starter, 3 minutes. Look at the mark allocation each time for the questions. I've started off with an easier question, right, one that you should be accustomed to. Okay, I'm going to give you 3 minutes. You don't need to rush through it. Let's start off nice and slow. 3 minutes, and your time starts now. How did you guys do? Let's see how it went. I'll go through it now with you. Remember, lots of this work you did at the beginning of the year, so you're going to need to go over it again, all right, just to get all those memories flowing and things coming back to you. Right, before us, as you can see in front, you should have recognized that there are two diagrams of a cell, and I will go through the questions with you. But what you should have noticed, first of all, that cell X was an animal cell, and plant cell, cell Y, was a plant cell. Right, so for the first question, right, they asked you which cell, X or Y, represents a plant cell. If we go back to the diagram, we would have seen that the cell would have been Y. And all you had to do, right, all they asked you to do was to say X or Y. And that is your answer that you're going to do for 1 mark. Now, question 2, there's a very important word in this question that you need to be able to understand, and that is the word visible. Okay? It says give 2, and we only 2. You know the guidelines, you may only do 2. Right? If you do any more than that, and the one is incorrect, you're not going to get your answer. Visible. What does visible mean? When I go back to my diagram, my differences must be seen in this diagram. So when we look at the differences, what do we notice? Always go for the more obvious. What do we see between these two? Don't go what you don't see. There are a few that you could have gone. Number 1 would have been the obvious, right? Cell wall is present. Another one would have been that a plant cell has a chloroplast. Okay, so let's go cell wall, right? A chloroplast. A large vacuole, because have a look over here, right? Animal cells also have a vacuole, right? A large vacuole would have been your reason. And from this point of view over here, did you notice your plant cell had centrioles? So you could have actually said from here that it's a plant cell because the centrioles are absent. Okay, so when we go to our back to our notes, okay guys, you only had to do 2 visible reasons. There were many. Right, there were many. You could, but only do 2. You are wasting time if you keep on listing as many as you can, hoping that you're going to get marks for it. Okay, so I'm going to put you on the diagram because that's where we labeled it. Then question 3, label parts A and label parts B. Now, if you have a look, part A is over here, and whenever we see this characteristic worm-like shape, can you see that? Always looks like a worm inside. That is always the mitochondrion. Okay, the mitochondrion. Number B, and this you sometimes get confused. Number B is leading from the nucleus, and it's those tubular passageways, right? And that is the endoplasmic. Let me move down a bit. Plasmic reticulum, which you are more than welcome to abbreviate as ER. It is a recognized abbreviation. Okay, the next question, right, the next question. Structure C plays an important role in cell Y. List 3 functions of the structure. So let's go back and see what it is. Number C is the vacuole. All right, now a vacuole has far more different importances than it might have in a plant cell than it does in an animal cell. So let's think. We need to think of 3. Right, we need to think of 3. Number 1, what does it do? The vacuole, right, write your vacuole first. Because sometimes if you write vacuole, it kind of focuses your attention on what you actually need to do. And it also shows the teacher that you know what the structure is. Okay, so it lists 3 functions. A vacuole, one of its main functions is storage. Right, inside there is cell sap, and inside the cell sap is water. With that storage, right, what do we also get? We get this word called turgidity. Turgidity. There we go, my spelling. Now, turgidity means, right, when water comes into the cell, remember, and it goes into the vacuole. What does it do to the vacuole? Right, it swells up. And in swelling up, then we call that the cell has got turgid. Right, another word that we can use, and we can use it here as well, right, a vacuole is for support. A vacuole in a plant plays far more a role of keeping the plant upright, right? Because remember, plant doesn't have a skeleton. So that's going to be the vacuole's role, to keeping it upright. Now, number 5, name the organic substances. Remember what your organic substances were: carbohydrates, proteins, lipids, enzymes, those kind of things. It says in plants, part number D. So we go to number D, right, and we notice number D, right, was one of the things was my cell wall. Now, my cell wall, the organic compound in my cell wall, I hope you got it, was cellulose. It's the part of the cell wall that we cannot digest. I hope you guys, all right, found that warm-up question a bit easier. We're now going to carry on to the next question. Okay, this sometimes can be a little bit of a difficult section. Please, all right, you need to know the different phases. You need to know what order the phases are occurring. You need to be able to identify, in other words, how do you know it's that phase? Because it's got these identifying characteristics. Let's quickly do these questions, right? We'll do them together and we'll see how well you did. At the bottom here, it says, study the diagrams below which represent the different phases of mitosis. You haven't done the other cell division yet, so you would only know mitosis. Okay, the first question is, label structures A, B, D, and E. Now I'm going to label it on the diagram. Now, sometimes the words get very confusing here. A, looking here, look at this whole structure, that's a chromosome. Now, number A is a centromere. Number B is one of the arms of a chromosome, which is a chromatid. Okay, let's see what other ones we needed to do. D and E. Now D is, if you have a look here, it's inside the nucleus. So number D is my nucleolus. And lastly, number E, you will see there is a pair of them. There are 2. Those are going to be my centrioles. Remember, my centrioles are always 2 structures, organelles, that lie at right angles to each other. One will always go to the one pole and one to the other to produce these spindle fibers. So be able to know your terminology when it comes to, right, mitosis. The second question, right, is by making use of numbers only. Guys, as soon as I've put it in capital letters, that is a very strict instruction, right? Numbers only. So if you give me the phases, I can mark it wrong, right? You're actually going to get 0 for it. So let's start. So it's for 5 marks. It's 5 marks. So we need to have 5 numbers. So let's have a look, right? And let's see which order we can put them on. Number 1 is interphase. In interphase, I need to always see the cell looks like it's whole and it hasn't started to divide yet. Right, so that would be number 1. Number 2 is, so interphase. Number 2 is prophase. What happens at prophase? I start to see my chromosomes, the nuclear membrane starts to disappear, and my centrioles begin to form. So this diagram is number 2. So far, all right. Then the third one, what comes after prophase? Metaphase. M is for middle, right? And here they all are on the middle, in the middle on the equator. That is diagram number 3. Okay, we're going to put all the numbers into the order just now. Once they're in the middle, then what happens? They start to move to the poles, right? So that is number 4. When the cell divides, that will then be number 5. Okay, so now you've got the order, but look at the question. Okay, sorry. The question said, make use of numbers only, arrange the phases into the correct sequence. So, which one was first? Number 4, then number 1, then number 5, then number 2, and then number 3. Okay, so the order, so I just arranged it in order. You now needed to go to the given number and you needed to put that in. Okay, the next question, same thing. Right, write down the number of chromosomes in a daughter cell at the end of the process. Now remember, the chromosome number must stay identical. So if I started with 1, 2, 3, 4, at the end in the daughter cell, this is a daughter cell, how many must I have? 1, 2, 3, 4. Okay, so how many chromosomes did I have at the end? 4. All right, they said in a daughter cell, not in both of them. So we had to keep the same number. Okay, next question. State one difference between a plant and an animal cell. Guys, if you have a look at this question, in this diagram, right? This diagram, this would be your difference. Number C. What happens in a plant cell that an animal cell doesn't have? Right, we have to form a cell plate. Right, what a cell plate has to form, right? Because what happens, right, we have a cell wall. And also, right, when we look at a plant cell, you can't always see it here. Okay, plant cells, we can't see any centrioles. Right, the centrioles are not visible. So that's how sometimes we know, even looking at the shape, that will give it to us. And the last question, right, on this is one I find that sometimes you don't understand what the question is. Why is mitosis biologically important? Biologically important means, why do we have to have mitosis? What is the importance for mitosis? And right in the beginning, what is mitosis? Mitosis is cells dividing, right? And why do cells divide? Why do cells divide? To grow, okay, to repair and replace. Right, what else we had? Asexual reproduction. So when we look at biologically important, what are we actually looking at? Why does it need to occur? Okay, 2 questions down. We need to take a quick break. And as soon as you have a nice big stretch and just get all those muscles moving, right, we'll come right back. All right, guys, welcome back. Stretch, maybe add a bit of water, or some of you might need coffee to stay awake. Right, guys, at this time of the exams, you know you should be eating properly, right? And if possible, ask mom or dad to maybe go and get you vitamin B tablets to help with your energy levels. I don't want to see you, all right, drinking all those terrible drinks, those energy drinks that are very bad for you. So around about exam times, you should be eating nice, lots of vitamin C to keep all the sick little bugs away, right? And vitamin B, right, to get your energy going. The next question, all right, is, I want to say it's a little bit more difficult than the ones that we have done. There is a bit of reading that I need to do, so you just need to bear with me. Okay. The question is, an experiment was conducted to demonstrate the movement of water into and out of a cell. As soon as I've said those, all right, that sentence, right, warning bells or ideas of what the question should be about should be going off. Okay, now this is how we did the experiment. 2 eggs, okay, we took 2 eggs, and what we did was we placed them in vinegar for a few hours to remove the eggshells. Okay, now if you have a look at the diagram on the next page, right, you will see, you can have a look of what we did. Okay, so what we wanted to do was we took the eggshells off, because if you underneath the eggshells, next time you go and have an egg at home, right, you will see there is the membranes. All right, once you take off the shell, there's like a white little membrane underneath. And that membrane is semi-permeable. Okay, and by semi-permeable, all right, it allows certain things in and certain things out. But as I said to you from the beginning, this word over here should have triggered things. And I'm not going to say it yet. I'm hoping that you follow with the question. Okay, now we've got these eggs, and all they have are these membranes. Okay, so now we take one egg. I'm going to go to the diagram. Okay, we take one egg, and we put the egg here, and only water around it. The other one, all right, in my beaker, another egg, which I've taken the shell off, and I've put it in a concentrated sugar solution. And I keep it there for 2 days. And after 2 days, this is what I observe, right? Observe means what I see has happened. The egg in beaker A has sunk to the bottom, but the egg in beaker B is now floating on the top. Right, let's look at our questions and see where those questions are going to lead us. Just note here, the sugar cannot move into or out of the egg. All right, the sugar solution stays the same. Let's go back to our aim right at the top. What did we want to see? The movement of water, right? The movement of water. So, name the process that is being investigated here. And that is why we had all these little dots. The movement of water into and out of a cell means link, 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 link. The movement of water across a semi-permeable membrane. All right, and I hope all your, I know, and all of you, the first thing I hope you two were thinking, all right, is osmosis. So if you thought of osmosis, we know that we are on the right track. Okay, now we have to go a little bit more in depth. Question number 2, now the mark allocation is slightly higher. If we go back to diagram number A, the egg, all right, sank to the bottom. So we put it in the water and it sank. Now the question they asked you is, explain why the egg in the diagram sank. Now, if I'm looking at a beaker over here, and I'm looking at water, and here I'm looking at an egg. And what's inside the egg? All your white and your yellow. So you've got little things dissolved inside. So now, what is osmosis? Osmosis is the movement of water. All right, so let's remember. Let me just quickly. MVT means water. Movement of water. Where from? A high concentration to a low concentration. Okay. Across a semi-permeable membrane. That's fine. So now why did the egg sink? The egg sank because water moves, now state that, moves from an area of high concentration, or you can use other words like high water potential, okay, in the beaker. That is important. Please tell me where it moved from. So water moves from an area of high concentration in the beaker. This was here. Let's make that nice and blue. This is where all the water is. This is where it was lots and lots of water. Water must move from here into the egg, right? So what does it do? In the beaker, into the egg. Now, there we go. Water moves from the beaker into the egg. So, what happens now I've got water? The egg is heavier because now it's filled with water. And now that it's heavier, what's it going to do? It's going to sink. Right, it's going to sink. And that is why, right, it landed at the bottom. So I hope you can then guess that the next question was, why did the egg in diagram B float after 2 days? Right here, we've got a beaker there. And now, instead of water, all right, what have we got? We've got this sugar. Those of you who like lots and lots and lots of things, and there's my egg. Now, where does water move? Water's still going to move from an area of high concentration to low concentration. Can you now, this is full of sugar, right? So this now becomes hypertonic. This now becomes hypotonic. Hypermarket, lots of things in it. Hypotonic. So where is my high water concentration? It is outside of the cell. So water moves, again, same thing, from a high concentration in the egg. All right, now it's different. The high concentration is in the egg, and the water is going to move into the surrounding sugar. So what happens to the egg? Right? It is less heavier. And what is it going to do? It's going to float up to the top. Okay? It's going to float up to the top. Right? Because what happened to all the water? Water moved out into the beaker. Right, so now all of a sudden the egg loses a lot of its water because all that white in the egg, the water, right, because we got it in the sugar solution, right, and water moves from high to low, so out of the egg into the sugar. Now the egg doesn't weigh as much, and when it doesn't weigh as much, it can float to the top. Okay, remember saying, some of you might have said moves from a high to a low concentration. That's fine, but where? From the beaker into the egg, or from the egg into the beaker. It's always going to move across the cell membrane. So be specific as to where the movement occurs. From the beaker into the egg, or into the egg from the beaker. All right, that shows you understand where the movement is going to occur. Okay, now the question was, explain why the egg membrane did not allow the sugar to pass through. Okay, now a membrane is semi-permeable. And what does that mean? All right, it only allows certain substances through, and usually most of the substances, right, are decided on, not say weight, on size. So why would the sugar not have gone through? Because it is too large. That's your 1 mark. To pass through the membrane. Okay, lots of you would have said maybe just too large. Too large for what? Too large to fly, too large to set sail, right? They are too large, right? Or they're too big to go through the membrane. And that is why they stayed on that side. Okay, the last question for this one, I did actually give this to my class as a test, and this they didn't do very well at, this one question. Listen carefully. What would happen if the egg in the diagram was placed in water that was heated to 35 degrees? Now, most of mine said that the egg would cook, all right? No. To cook, you need to boil, and boiling occurs at 100 degrees. So guys, 35 degrees, that is your body temperature. All right, so that is normal. It's actually normal heat. Okay, so what happens when we heat something? Right, when we heat something, 35 degrees is not very hot. Right, if I touch myself, I'm maybe slightly warm, but I'm not on fire. I'm not going to burn everything. I'm not going to denature everything inside there. If I slowly start to warm, all right, what's going to happen to my process? Heat speeds right, the process up. So what's going to happen? It's going to happen quicker, right, because the heat has more energy. So my process is going to happen quicker. All right, so guys, boiling 100 degrees. Now, going on to our enzymes, again, I've put a simplish diagram in, but one that you need to know. It's a very common diagram when it comes to the enzymes, and it is my, right, it is my lock and key. Okay, question over here. So here we have two, all right, a couple of diagrams, and let's see if we can identify, all right, what the answer that we need to answer. Okay, so what I'm going to do is, right, we're almost ready for another break. I'm going to give you 2 minutes just to quickly jot down 2 minutes, right, and then we come back, and then we'll see if we need to have a little quick break or not. So 2 minutes, your time starting now. Hope you guys did all right. Okay, what we're going to do now is we're going to take a quick little break. So if you didn't have time to finish, we'll give you a little bit of extra time. When we come back, we'll mark, and then we'll carry on with the rest. Okay, quick break. Again, stretch or quickly finish. See you now. Okay, welcome back after that little stretch. Get the shoulders going, everything moving again. Right, let's quickly do the answers to the questions that we did on the enzymes. Right, when you looked at the diagram, as I said to you, I hope the first thing you see is the lock and key. Right, that was the whole concept that we look at when we look at the enzymes. Let's go through the possible answers. What is an enzyme? Now remember, you are talking in biology. Okay, now when we talk in biology, we're not talking about science. Okay, so an enzyme is a biological catalyst, and it speeds up reactions in an organism. All right, you could even say it in your body. All right, now the next question, number 2, right, we have to provide labels, all right, for structures A to D. So let's go back. I'm going to label it on the diagram. It's much easier than having to go back and forth. Now, what you had to describe here was you had to separate which is the enzyme and which is the substrate. See the words that I'm using? Okay, enzyme, substrate, complex. All right, so now an enzyme, one of the properties of an enzyme is it doesn't get broken up. Okay, so it doesn't change. So if you're not sure which one it is, always keep saying enzymes don't change. So if we look at the diagram, right, which is the one that stays the same is this one. So this is an enzyme. What it works on is a substrate. I can hear voices in my head. Oh my, yeah. Substrate. Right, number C is this whole thing over here, okay, which is the enzyme substrate complex. Right, that is when the enzyme and the substrate bond together. Number D over here is the process, the products. Okay, number D is the products. So when we look at enzyme action, we have an enzyme, it works on something, and at the end, that something needs to be changed. So there's your A, B, your C, and your D. Guys, those are very common things that need to be asked. Those are the basics that you need to know when it comes to the enzymes. Okay, our next diagram, as I said, I'm going to put here on diagram, just in case you have to go back to it. List 2 characteristics of enzymes. And if you had listened to me just now, you would have heard what I said. Enzymes, there are a few characteristics. What are they? All right, number 1, they can be reused. Right, so we know that the enzymes, we have a look there, right, they don't get broken down. Okay, what else? They are substrate specific. They will only work on certain, certain substances. What do we also know? They are temperature specific. Right, certain enzymes will only work at certain temperatures. They are also pH specific. And when it comes to these things, when we need to know them, because we need to use that, right, when we do experiments. Because we can use these principles when we do experiments. We can experiment changing the temperature, changing, the pH, changing what it works on, and seeing if it's going to work or not. Right, question number 4, you needed to think a little bit here. This was your in-depth question. It says enzymes that break down fats and proteins are often added to washing powders. Okay, give 3 reasons why the addition of enzymes make the powder more effective. Now, what does an enzyme do? Fats and proteins are organic molecules. Right, so they have they're food, right? And what they do to clothes is they stain. So why do we want to use a washing powder? Right, because a washing powder is an enzyme. Give 3 reasons why the additions of enzymes make the washing powder more effective. Because the washing powder is now an enzyme. So what does the washing powder do? Right, for starters, an enzyme needs to work in water. So when we wash our clothes, what do? We've got water and we use the washing powder to it. So why is it more effective? Number 1, it's going to break down the stain. I'll put this stain, the stain, let's put there. Let's take let's get back. Okay, we have a stain. So what's it going to do? The enzymes are going to work on it, and what does the water do? Right, the enzymes break down the fats and proteins. And in breaking down the fats and the proteins, what do? They take the stain away. Right. And what is staining away? They are cleaning. Right. So why do we add enzymes? To break down into smaller particles, right? And then what do the enzymes do? They break it down, right? And then what's going to happen is all those broken down foods are going to dissolve in the water, and the water is going to wash it away, and that's how we're going to remove our stains. Okay, you didn't think that, right? Enzymes were washing powder was enzymes. Right, guys, the next section you can see is a question, and I know for me, this is the one that my kids battle with the most, trying to see which kind of mammalian tissue is which. Let's have a look at the diagrams to see if we can look at familiar things. The question below says, look at the diagrams below and answer the questions that follow. Okay, you have got three tissues in front of you, and now we're going to need to answer questions on each one of them. Right, the first thing you are asked is to identify the tissues labeled A to C. And I'm going to go back to the diagram where we are going to do it. Guys, usually they are going to use the more obvious ones. Bone would be number A, right, because of the knife, remember, those round circles. Number B, can you see these little cells in the hollows with no fibers? Right, that is hyaline cartilage. Right, and if you have a look over here, this is sometimes where things get a little bit tricky, right, because it is muscle. Now, when it gets to muscle, you sometimes can't tell the difference between, all right, skeletal muscle or cardiac muscle. But if you have a look, do you notice at some places, right, there is no, so there's a bit of matrix, and at other places they have these bridges, right? They're not just single strands, they have these intercalated discs. And that would have been cardiac muscle. Right, cardiac muscle. I know they sometimes, for you, it's very difficult for you. Give one function of tissue number A. Tissue number A is bone. So you could have quite a few functions here. All right, protection is the first one that comes to mind. Bone also strengthens. When we talk about bone, it also produces blood cells. So there's a few that you could have thought of, but those two are usually the most common. Now, question number 3, okay, is how does the matrix of tissue A differ from the matrix of tissue B? Okay, very nice question that, a bit of your higher order question. Your matrix is your background. Now, if I were to color in the matrix, that would be my pink. Do you see all the white here? That is the background. For the hyaline cartilage, it is all of this. Now, when we get to the differences, right, number A, let's start off by saying, oopsie, going the wrong way. Let's start off by putting what number A is. Number A is bone. Number B is hyaline cartilage. Right, so what is the difference between the two? The matrix, the background. Bone is hard. So, why is that? Because of the calcium, right, and the collagen, and all the other salts. Right, what are they? They're in crystal form. And when they're in crystal form, or they're hard in form, right, they are hard. So my matrix is hard, right? Whereas hyaline cartilage, it doesn't have a hard matrix. The matrix actually is collagenous fibers in a matrix that is semi-solid. Right, so what do I say here when I have a semi-solid? Right, I can almost, it's not hard. Right, the matrix, it's not hard. So one is collagen fibers. That collagen, right, even though I find collagen in bone, the rest of my matrix is all hard. Whereas in the hyaline cartilage, even though I can't see those fibers, they are embedded in the matrix. And what does that give? It makes it quite hard, hard-ish, right? As I say, semi-solid or semi-hard. It makes it more flexible, right? It's not as hard because when it comes to hyaline cartilage, we need to be more flexible. It needs to be able to have a little bit of bend. Okay, so that's what we mean when we talk about the matrix in tissue A, right, and in tissue B. Okay, next question. All right, next question. Where in the respiratory system would you find tissue B? Now, a respiratory system, right, that is your nose, your gaseous exchange system. So it's your nose and it's going down your throat, right, into there. So where would you find them? Now this, even though you haven't done the respiratory system, when you did the mammalian tissues, what did you do? You were told that hyaline cartilage is found where? And hyaline cartilage in your trachea, right there are C-shaped cartilaginous, all right, tubes. So that's one place where they're found. They're found in the trachea, right, the tubes. And also when you swallow food, all right, and here's the food at the back of your throat, before it goes, the trachea is in front and the esophagus is at the back. And when you swallow food, you've got to make sure that the food goes into that tube and not into the trachea. And over there, we've got a nice little hyaline cartilage clip, all right, called your epiglottis. All right, so in the respiratory system, that is where it's found. Okay, there we go, making up in back of your throat there, and in keeping the tubes open. Now, give the function of tissue B in the respiratory system. Now, the function of tissue B, there we go, is to keep the trachea open. All right, so that would be the function, and the epiglottis's function would then be, okay, to stop food going into trachea when we swallow. Okay, so when we're looking at our respiratory system is our gaseous exchange, our tubes going into our lungs, and that is where we're going to find the cartilage. Okay, next question. Now, question number 5 is, in which part of the breathing system would you find tissue C? Now, when we talk about the breathing system, tissue C, all right, is going to be our cardiac muscle. And when we say the breathing system, right, we actually mean the cardiovascular system. So in which part of and that is going to be in the heart. And why do we say it's in the breathing system? Is because in breathing, we need to get oxygen in. Right, and when we breathe oxygen in, that oxygen has to take that oxygen everywhere in the body. And I'm looking at the time and I see our time is up. All right, guys, thank you for joining today. We'll see you next week. We will look at more questions on paper 2. See you next time. Bye.